Hello class, today we will discuss about gene therapy. But before that, I would like to test your concepts of genes and genetic mutation. So, please one of you, tell us about genes and genetic mutation.
Hello everyone. I would love to explain everyone what are genes. The human body is made up of cells. If you look inside the cell, here is the nucleus, where your DNA is present. DNA is the genetic material which stores the genetic information. It is made up of nucleotides. While the segment of the DNA sequence present in the cell specified for a particular feature or function is called a gene. Any change or alteration in the gene or a DNA nucleotide is called genetic mutation. It can affect the body functionality. Different colors of eyes is one such example of genetic mutation. This condition is called heterochromia. Well, these mutations are not affecting health. But there are other genetic mutations that affect the overall health of the body. Hence these disorders are called genetic disorders. One good example is cancers.
Thank you for good and concise explanation. So, getting back to our topic.
Gene therapy is a genetic remedy for illnesses that includes changing genes inside your body's cells to be able to treat or cure the disorder. It replaces or inactivates diseases inflicting genes. It also involves the advent of new genes into the body to deal with a particular disease. Adenosine deaminase (ADA) deficiency is a congenital disorder which weakens the body's immunity and capacity to fight back. In 1990, for the first time, a 4-year-old patient, who was diagnosed with ADA deficiency, treated with gene therapy. Drive W. French Anderson took her white blood cells, inserted genes that produce ADA, and the corrected cells have been injected back into her body.
Is it safe procedure? Or it has some risks as well?
The incorporated risk factors is the reason why it is still not become an effective medical procedure. One such risk is that, in gene therapy, when the corrected gene is introduced via an agent or a vector, such as viruses, there is a chance that it can lead to organ failure as the body won't accept the entry of foreign cells. Mechanisms of gene therapy include the replacement of a disease-causing gene with a healthy copy, inactivation of the disease-causing gene, and introduction of a new gene into the body. So, that is it for today. I want you all to explore more about gene therapy.
Today's lecture was great. However, I'm also curious to know more.
Hey, I found that gene editing techniques like CRISPR-Cas can be used to reduce the risk factors we have studied previously. CRISPR-Cas system is a gene editing tool. CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Sequence. The Cas is the CRISPR-associated protein, which has a significant role in editing the DNA sequence.
It's nice. From where do you get this information?
I found this interesting article with easy-to-understand information on ScienceCept's website. We can also read about cancers and CRISPR-Cas here.